In this series of videos, we're going to be studying the pyrimidine synthesis pathway. So we're going to begin by discussing the pyrimidine synthesis pathway in more detail, and then we're going to move on to discuss the role of folate and B12 in pyrimidine synthesis, and then towards the end we're going to discuss the clinical correlates to the pyrimidine synthesis pathway, and we're going to talk about the pathologies that happen due to abnormalities in this pathway, and also the drugs that we use that target the pyrimidine pathway. Okay, so the goal of the pyrimidine synthesis pathway is to synthesize the three pyrimidine nucleotides, and they are uridine, thymidine, and cytidine. The first part of this pathway is going to be the formation of uridine, and uridine is going to be used as a template to make thymidine and cytidine. The best way to understand the pyrimidine synthesis pathway is to study the formation of each pyrimidine nucleotide separately. Okay, so we're going to start by studying the synthesis of uridine or uridine monophosphate. So we're going to start with the amino acid glutamine, and glutamine is going to be converted into carbamyl phosphate by the enzyme carbamyl phosphate synthase. Then we take the carbamyl phosphate and we convert it to orotic acid. This step is going to be catalyzed by the enzyme dihydroorotate dehydrogenase, and we're going to consume an aspartate amino acid in this process. Orotic acid is then going to be combined with PRPP to form UMP, and this process is going to be catalyzed by the enzyme UMP synthase. Just to recap what we just studied, so we're going to start out with glutamine. Glutamine is going to become carbamyl phosphate. Carbamyl phosphate is going to be converted to orotic acid. Orotic acid is going to be combined with PRPP to form UMP. And remember we said that UMP is going to be used as the template to make the other two pyrimidine nucleotides, and they are cytidine and thymidine. The next thing that we're going to discuss is the synthesis of cytidine. So as we said before, we're going to use UMP as a template for the synthesis of the other pyrimidine nucleotides. UMP is going to be converted into UTP, so uridine triphosphate. So basically we're just going to add two phosphates. And then UTP is going to be converted to CTP. So what we did is that we changed uracil into cytosine, and we formed CTP, or cytidine. So the final pyrimidine nucleotide that we have to synthesize is thymidine. So like we said before, we're going to start with the uridine monophosphate base pair, and that's how we're going to synthesize thymidine. So something really important to know is that thymidine is only found in DNA. That means that we only have to synthesize the deoxy form of thymidine. Now remember, the difference between DNA and RNA is that DNA does not have an OH group on the second carbon, while RNA does have an OH group. So this is why we only have to synthesize the deoxy form of thymidine, because it's only found in DNA. Okay, so like we said, we're going to start with the template uridine monophosphate, and we're going to convert it into uridine diphosphate. So we're just going to add a phosphate group. The next step is going to be to convert the uridine diphosphate into a deoxyuridine diphosphate, and this is catalyzed by the enzyme ribonucleotide reductase. So what happens here is that we just remove this OH group to form the deoxy form of uridine diphosphate. The next thing is that the deoxyuridine diphosphate is going to be converted back into the deoxyuridine monophosphate. So we're gonna remove the phosphate, and then deoxyuridine monophosphate is going to be converted into deoxythymidine monophosphate by the enzyme thymidylate synthase. So when we were talking about pyrimidine synthesis, we said that deoxy-UMP is converted to deoxy-TMP by the enzyme thymidylate synthetase. Now, the difference between uracil and thymine is only one carbon, so thymine contains one extra carbon over uracil, and that's going to be the difference between uridine and thymidine. So where does basically thymidylate synthase get that extra carbon from? It gets it from the molecule N5-N10 tetrahydrofolate. So folate is going to be the source of the carbon that helps us form thymidine. So folate in humans comes from our diet, so we ingest it in our diet, and it's converted in the body to dihydrofolate. Dihydrofolate is then converted to tetrahydrofolate by the enzyme dihydrofolate reductase, and then tetrahydrofolate is converted to N5-N10 tetrahydrofolate. That provides the carbon in order to make thymidine. So we just talked about the importance of maintaining adequate levels of N5-N10 tetrahydrofolate for the synthesis of thymidine. Okay, so what is vitamin B12's role in this process? Well, sometimes N5-N10 tetrahydrofolate can acquire a methyl group and become N5-methyl tetrahydrofolate, and of course this becomes useless in the body. So it's vitamin B12's job to take that extra methyl group and convert N5-methyl tetrahydrofolate to tetrahydrofolate. tetrahydrofolate is then converted back to N5N10 tetrahydrofolate, and that maintains adequate levels of N5N10 tetrahydrofolate. So as we can see, vitamin B10 is very important in pyrimidine synthesis.